All right, ladies and gentlemen, so we are going to wrap up chapter 27, really kind of just looking at some of the crucial roles that prokaryotes play in our biosphere, in our ecosystems. Um, First and foremost, just go back to the fact that they play major roles in our chemical recycling. So, going through those biogeochemical cycles, they all, in some space, contain prokaryotes as part of the recycling of nutrients. Two of the big ones are the carbon cycle and the nitrogen cycle. So when we look at your carbon cycle, you've got not only larger organisms doing things like cellular respiration and photosynthesis, um, but prokaryotes also play those roles in breaking down um, carbon dioxide and turning it into sugars and vice versa, restoring oxygen to the atmosphere via cyanobacteria doing photosynthetic processes. And then just remember that all of that carbon ultimately has to get decomposed and returned back to the soil to be taken up again as well. So you've got a lot of decomposers that are prokaryotic organisms playing a role in that carbon cycle too. Nitrogen cycle, also some decomposition just needing to happen there in order to break down organisms and unlock not only the carbon but the nitrogen and other elements as well. And remember that your nitrogen fixers, taking that atmospheric nitrogen and turning it into nitrogen compounds that other organisms are actually capable of using, are all prokaryotes. So they need to be able to convert that nitrogen gas into those nitrogen compounds in the soil and in water, for plants to use, and then for that to move up the food chain from there. Prokaryotes really fill the role of all sorts of symbiotic relationships. Remember that symbiosis is just that ecological relationship between organisms of different species that are in direct contact. And you have prokaryotes that fall into those different types of symbiosis. The mutualism, where you've got both participants benefiting, commensalism, so one organism benefits, but the other really isn't helped or harmed, at least not that we're capable of discerning at this point. And then those parasitic relationships, where that parasite is benefiting at the expense of its host. So when we look at some examples of mutualistic prokaryotes, just a couple examples. So we go down to those sort of deep ocean layers, and you have bacteria that are capable of bioluminescing, and they live within the flesh of fish species and things that live down there. This is an example of the flashlight fish, and it actually bioluminesces, but it's not because it's producing it, but instead it lives mutualistically with bacterial colonies. Those bacteria bioluminesce as part of their metabolic processes. The fish provide those nutrients from the fish provide nutrients to the bacteria, I should say, and then that bioluminescent relationship, that capacity that those bacteria have to create that light, allows the fish then to attract its prey as well as its mates. So both species are benefiting as a result of that relationship. If we look at our human intestinal tract, our digestive system, it's estimated anywhere from 500 to 1,000 different species exist mutualistically with us, as well as many other organisms that have these digestive systems. And those bacteria have co-evolved with us to digest food that we wouldn't naturally have enzymes to produce ourselves and break down ourselves. So in the process of digesting that food, the bacteria take in some of the nutrients and things that they need from us. But it's beneficial in the sense that they are also breaking things down and allow we are able to absorb those nutrients then as well. Um, Most kids will think of the pathogenic prokaryotes first off, right? So about half of all human diseases are caused by some sort of prokaryotic species. One of the most widespread in the US, and also in Europe, is Lyme disease. So this is caused by a spirochete that exists and lives in the guts of these ticks. Those ticks then bite us. In the process of them feeding on that blood from us, they will eventually regurgitate some of their gut content, that contains these spirochetes if they are in fact infected with it, and that leads to all of the complications that we end up seeing with Lyme disease. So most commonly, you get this sort of bullseye rash. Um, those bullseye rashes can be at the site of the bite, but they don't have to be. They often will occur in places like the back of your knees, in your armpits, places where you have copious amounts of lymph nodes. That seems to be a place where these rashes congregate. Um, Not everybody notices the rash, but the rash does occur, we think, in about 80% of cases. Um, because they're in sort of awkward places, you may not even notice that they are there, but they are. And that's usually the one big telltale sign of Lyme disease, because we don't really see that rash with any other disease out there, which is kind of crazy. A lot of the prokaryotes will actually cause illness not because they themselves are in your body reproducing, but it's the things that they are creating as a result of their either their metabolism or something that they sort of release when they die. Um, so a lot of pathogenic prokaryotes produce these toxins. If they're exotoxins, these are proteins, these are molecules that the prokaryotes are producing as a part of their metabolic processes, and they're releasing them. They're sort of waste products. Um, and it's actually not the bacteria itself that is causing the symptoms, but it's the release of those exotoxins that causes the symptoms within a infected patient. So things like cholera, botulism, these are going to cause those symptoms that we typically see. Diarrhea, intestinal issues, that kind of thing, are often caused from these two species. And the reason we know it's the toxin and not the bacteria making you sick is because even in the absence of the actual prokaryote, you still have those toxins present and you're still showing symptoms. Endotoxins are actually produced as a toxic component of the outer membrane of a handful of gram-negative bacteria. And these only get released when the bacteria is killed off. Salmonella, for example, is one of these. So we go treating you with antibiotics, or your body natural defenses try taking care of that bacterial uh, pathogen, and you actually end up seeing a worsening in symptoms before you see an improvement. And that's because the bacteria are releasing these endotoxins as they die. Your body's got to metabolize and break that down, or we have to treat you in some other way to take care of those endotoxins before you end up feeling recovered from it, even though the bacteria aren't actively growing. And we look at prokaryotes in terms of research and technology, and they are certainly a hot topic when we talk about things like bioterrorism. Um, there are major entities, government entities, who have bacteria kind of on file, if you want to think about it like that. They are basically in deep freeze, and they're there supposed to be for research purposes, or if something were to, what we thought was eradicated, all of a sudden came surging back in the environment. We want to be able to have these as a way to study them for future disease processes. But they also are then potentially capable of being weaponized. Anthrax was one that was a big concern after 9/11. It was showing up in mail to high government officials, and we think perhaps there was some connection there. So you breathe in these anthrax spores. They were mailing them in letters. You open the letter, it puffed out this powder that were these anthrax spores. You breathe them in, and you end up contracting um, the disease as a result. Um, Botulism is another one. Plague, right? So could all of these things potentially be weaponized in the sense of growing stronger strains, resistant strains, this, that, and the other thing? Sure. How much of that is happening? I don't know. You could really start getting down the rabbit hole of conspiracy theories and this, that, and the other thing. Um, Whether there's truth to that or not, who knows? But the potential is there for sure. On to happier topics. We use a lot of prokaryotes in agricultural processes, in production of foods. Um, we look at something like the Lactococcus lactosci. These are ones that we use a lot in cheeses, okay, along with a bunch of fungi that also play roles in producing cheese. The lactobacillus and some of the subgroups there are ones that are common in producing yogurts. So we'll take that typical milk, treat it with these different bacterial cells, and be able to create products out of them, which is kind of fun. So depending on the different species that you use and how long you let them sit there, you can get different textures out of it. You can get different tastes out of it. That sort of sharpness at the back of yogurt is a result of the acids that are produced from the lactobacillus going through fermentation. So it's kind of fun. And we definitely use them for biotechnology purposes. The fact that we have been able to take up the ability for bacterial species to do things like transformation and take plasmids out of their environment. We can genetically engineer those plasmids to now contain genes that we want. We also take advantage of restriction enzymes and being able to cut up the DNA, and those restriction enzymes are a natural product of prokaryotes. So we can modify these and create these plasmid rings with all sorts of genetic information to produce things like vitamins, antibiotics, hormones, you name it. So long as we have a genetic code for it, we can splice them together in these plasmids and go through a transformation reaction and get those bacteria to take up that genetic information. When we do that, we can then either use them to just sit there and make copies of that gene for us for certain purposes, or we can let those bacterial cells now create those products for us. The example I often use with the underclassmen is the production of insulin. So historically, insulin came from pig sources. Well, there's a whole host of complications that came from treating diabetes with insulin from pigs. Not only do you need to keep the pigs alive and be able to extract the insulin from them, which is incredibly time consuming. It's a lengthy process, it's an expensive process. Even just housing the pigs is expensive. Um, it also came with the fact that it wasn't human insulin, and so some people didn't take to it well. There were some religious and ethical concerns with it, just using pig product, period. If you are a very strict Jewish practitioner. Pigs aren't considered kosher, so what does that mean in terms of your ability to use that material, so on and so forth. So biotechnology, genetic engineering has kind of gotten around that problem because we now are capable of taking the human insulin gene out of a human cell, splicing it into a plasmid, putting that plasmid with the human insulin gene into a bacterial cell, and bacteria don't know any different. DNA is DNA is DNA. So they will use that genetic information to go through protein synthesis and produce human insulin. And it's faster. I can do it in a petri dish in a lab, and it's actually human insulin. And so the risk of complications from it coming from another species don't exist either, which is pretty cool. We do some other cloning and things. E. coli is kind of the common one, especially when we're talking about doing things within human species or other mammals, or just general gene cloning. E. coli is a really nice bacteria to work with from a lab standpoint because it's generally easy to grow. Most of the time, it's not pathogenic. It doesn't make us sick, so we're not worried about um, us taking in a little bit here and there if it happened to get to that point. It's not a huge bio safety issue. Um, so that is often the species of choice. Agrobacterium is another one that also gets used a lot. That is a bacteria that is pathogenic to plants. But if we genetically modify it, we can get it to still do its infection pathway, but instead of inserting its own genetic information and basically creating these big, like, tumors almost on the plants that it infects. We remove that ability from them and instead insert other traits that we want. So drought resistance, higher yield fruits, whatever the case might be. And that's a common, what we call vector to getting that genetic information into plants. We also use prokaryotes for bioremediation pieces. Um, there are some prokaryotes that are very good at being oil eaters. And so you get an oil spill and they can go out and spray that spill site with these prokaryotes that'll kind of eat up that bacteria, that oil, excuse me, clean up the problem. Um, Fertilizers, right, can also then be incorporated in that and speed up the growth and breakdown process of just native bacteria that may already exist. We're improving and hastening the cleanup process as a result. We definitely use anaerobic bacteria and archaea to help decompose organic material and things like the soils. Came across a project at Penn State a few years ago where they were using bacteria to take human waste sewage and convert it into something that would essentially be sort of clean, if you will, but that could get turned around and used as fertilizer. So the bacteria were taking the place of chemicals to basically sterilize the waste material from humans and turn around and use it as fertilizers on crops. It is being done even locally, but most of that human fertilizer isn't being used on anything that goes directly into human consumption crops. It's in materials that may be fed to animals or used in other sorts of endeavors. It's not anything, at least at this point, that I'm aware of that is approved for spray on human directly consumed human crops, if that makes sense.